Hi guys, welcome to our video on protein synthesis. So this is just another way we're going to kind of approach this. So kind of my guided notes, calling out some important details and things I want you to recognize about this process of making proteins inside the cells. In terms of big questions, number one, and this is our, put double stars next to this one, how is DNA's code expressed? We have come off the tail of looking at DNA structure and looking at replication as a process, and we really discussed how DNA is essentially storing information in those sequence of A's, T's, G's, and C's. And so we're going to look at now, how does that cell actually read and decode that information to convert it into a protein? What we're then going to do is look at mutation. So we're going to look at, well, how does changing the sequence of DNA actually have a phenotypic effect? So a visual, actual noticeable effect on the cell itself. And part two really here is actually the origin of genetic disease. So understanding why it is that someone can inherit and have some sort of pathology related to some genetic disease. Ultimately, at the end of this, I want to be able to give you guys a nucleotide sequence and have you guys be able to go through the whole process of protein synthesis and tell me what that protein structure would look like. In terms of where we're at visually, we want to remember that when we start talking about genes and we start talking about DNA, genes are actually just small sections of our chromosomes. So the vast majority of your DNA does not code for proteins. And we'll talk about kind of what makes up the vast majority of your DNA. But there are regions of it called genes. And if we zoom in on those genes, we end up with just a specific sort of finite sequence of A's, T's, G's, and C's that your cell has the ability to recognize and identify. And these sequences are what get used to actually figure out the sequence of amino acids that need to go in order to form the final protein the cell is trying to make. So this slide right here is just kind of a visual walkthrough of kind of where we're at right now. So we're talking about regions of a chromosome that code for a protein. You guys have seen this before. So this is the central dogma. So this is in biology, basically our understanding of sort of the molecular importance of DNA. And so again, dogma refers to sort of a foundational belief or understanding. And so we're going to talk about kind of what this means. But essentially, the central dogma in biology tells us that DNA gets converted into RNA through a process known as transcription. And we're going to talk a lot about transcription. If you look at the word itself, trans means across and script means to write. So what you're actually saying with this beautiful word right here is you're saying write across. And so what we're going to see in transcription when we do this a little more in depth is how that code in DNA, those A's, T's, G's, and C's, is essentially just going to get copied into RNA. So it's like we're going to make a little molecular photocopy of that genetic information that we need in the cell. The second half of the central dogma says that RNA is then converted into a protein, and that is called translation. And you guys know translations. You've taken foreign language classes before. So when you think of translation, you talk about going from one language to another. Well, in the second stage of this, that's essentially what we're going to do in a biological sense. We're going to go from the language of nucleic acids, so the language of A's, T's, G's, and C's of nucleotides, into the language of proteins. We're going to convert those A's, T's, G's, and C's into amino acids. And that's why that second phase is called translation. It's that conversion point between a nucleic acid and into a protein sequence. So these down here, transcription and translation, these are just the names for the different conversions that we're now going to study as we go through protein synthesis. A big idea for DNA, and hopefully this is familiar to us, is that DNA contains coding regions called genes. And genes are essentially the recipes to make a protein. As far as we know, one gene equals one protein, meaning that each gene that we have inside of our body codes for one protein. And proteins are essential molecules. We had kind of in our intro unit talked about sort of what they do. They regulate, they are structural, they allow for transport and movement. You literally cannot have a living thing without having proteins. And literally how the cell has the ability to make these proteins to keep themselves alive, to maintain homeostasis, to build themselves, is because the instructions for those are in their DNA. Now, this is kind of a throwback question to cell structure, which I know is kind of a middle school territory for many of us. So we are going to talk about how proteins are created. And we need to remember that when it comes to cells, cells have some organization when it comes to these processes. They have specialized structures that are going to allow them to create and perform essential functions. We find that all cells, this includes prokaryotic and eukaryotic cells, all cells have a specialized structure inside of them that actually assembles or is going to put proteins together during translation. And the name for that structure is a ribosome. So while we do not find a nucleus in every cell and we don't find a Golgi apparatus, and we don't find mitochondria in every cell, what we do find is that this job right here of making a protein, again, so fundamental to life that it does not matter if a cell is prokaryotic or eukaryotic, they all have ribosomes. They all need the ability to build their own proteins. We do have, so DNA we know is where we're going to start this story. So kind of picturing the central dogma in your head, DNA converting into RNA, RNA converting into protein. We're going to start with DNA. DNA we know is this master copy of all of the instructions to actually make a protein. But in eukaryotic organisms, we know that it's inside the cell. And so we end up with a little bit of kind of a logistics problems when it comes to eukaryotic protein synthesis. And that is, how do you actually get the information that is inside the cell, so inside the nucleus of the cell, to the ribosome? And so what we're going to have is an intermediary molecule. And this is kind of the first stage or transcription of protein synthesis. We see it kind of playing out here. DNA is going to get converted into RNA, and we are going to talk specifically about the type of RNA that gets converted into. But we notice that RNA plays kind of a huge supporting role. So if we were to cast this movie of protein synthesis, yeah, DNA is a star, protein synthesis is a star, but you cannot have this story play out without all of your essential supporting characters. And in this case, that's RNA. And there are three major types of RNA that are involved. There's what's known as mRNA, tRNA, and rRNA. Right now, we're going to walk through and talk about specifically how these different RNAs are involved. So the first one we get to is mRNA. And this is short for messenger RNA. The RNA should always be capitalized because it's an acronym for ribonucleic acid, and the M is always lowercase to identify it as messenger RNA. So as we said in our eukaryotic cells, the DNA is inside the nucleus, and DNA is also the master copy of all of your genetic information. You really don't want a cell kind of cutting up this information, moving it around a cell a lot, because it just creates a lot of chance for it to get damaged. And you would never want sort of the master copy of all of these instructions to be damaged in any way. So what the cell actually has is a little workaround. What it's going to do is essentially make a molecular photocopy of whatever gene it is trying to create a protein for. And it's going to do that in the form of an mRNA molecule. And this is what happens during transcription. We're going to talk about this process a little bit more. But essentially, what you have is your gene coding region. So the section of A's, T's, G's, and C's to make a protein. You're going to have a copy of that made in mRNA using complementary pairing. So it's going to borrow a lot of the similarities in terms of logistics and mechanics that we saw in DNA replication. But instead of creating two double-stranded identical DNA molecules in transcription, we're just going to create a single-stranded sort of strip or copy of a DNA sequence. So here we go, transcription. Oops. Oh, that's all right. I filled it in. So transcription right here, we're going to notice that in order to copy DNA into RNA, specifically mRNA, we're going to do some complementary pairing. So we are going to keep our first complementary pair from DNA replication. So if we remember, cytosine and guanine are complements. They always go together. But we notice that we have a little bit of a difference here. Whereas normally if we were in DNA replication, so looking at copying DNA to DNA, we would have adenine paired with thymine. We notice that because we are going to make this copy of a gene into RNA, RNA has a different base pair. It pairs adenine with uracil. Uracil is a less stable nucleotide than thymine. Now, for DNA, this would be bad. You would not want to have uracil inside DNA because remember, DNA is that master copy. But mRNA is just the photocopy. So what the cell will actually do is it'll use this readily available, so very cheaply available uracil in place of thymine in order to build this temporary photocopy, make the protein it needs, and then if it breaks down later, what does the cell care? It was a temporary copy anyway. And so uracil is the nucleotide that we're going to find in place of thymine inside your RNA. What this process actually looks like, and we'll watch lots of videos, we'll see this is represented in lots of different ways. This is a very simplistic model of it. Is let's imagine that you have this sequence in your DNA. So imagine that this is a very, very tiny gene. So you have ATTGCA. So these were your nucleotide sequences. This is the instructions the cell needs to figure out how to make a protein, but this is inside the nucleus. And so what the cell is essentially going to do is it's going to use those complementary pairing rules in order to make a copy of this code. So the A in this case is going to pair with, because remember we're turning it into RNA, So the A is going to pair with U. So we're going to have uracil. The T still pairs with A, because remember, adenine is still in your mRNA. It's just the T's that you wouldn't find in this sequence. So T pairs with A, T pairs with A, and then G and C, nobody touches that pair. They are DNA and RNA together for life. And so what we find is that G and C are still pairing together over here. Now, I can easily identify my top black strand as DNA because I see thymine, and I can easily identify my bottom strand as RNA because I see uracil. So it's an easy way to help you identify between them. Now, what we have to appreciate is that what you essentially have in that red sequence on the bottom right there is you have a molecular photocopy. This is now sort of like if I took your textbook and I run a copy off on like the machine, you now have something you can carry around, you can kind of write all over, you can treat maybe a little bit more poorly, but it has that same data in it. And that's kind of what the deal is with this red strand down here. The mRNA strand has essentially, through that complementary pairing, made a copy of that gene. And it now has that information, the instructions stored in it. But now this copy can leave the nucleus, it can travel around the cell. And if it gets damaged, that's okay. We can make another copy later on. So that is transcription. We want to make sure before we move on and talk about sort of what happens with this transcribed mRNA sequence that we appreciate and make sure that we are differentiating between replication. So if I were to ever, or if you saw an MCAS, a question that asked you to take that same sequence and replicate it instead of transcribe it, what sequence would you put underneath it? Good. So hopefully you were able to remember that when we are asked to replicate DNA, that's the process where we are using our complementary pairing A's with T's, G's with C's, because we're copying double-stranded DNA, making two identical copies of DNA. So in this case, you would have A and T pairing. Now, a quick and easy way to do this is if you look kind of the difference between your transcribed sequence and your replicated sequence is it really is just swapping out T's and U's. U's are found in RNA, T's are found in DNA. What I want you guys to do right now is take a quick second and I want you to practice those two problems on the bottom. We'll go over these together as a class when I see you guys next time. So pause here. And again, it has like a couple guided practices on the bottom for you to give it a shot. Good. So hopefully you've gone through and practiced that. All right. So what we're going to do right now is talk a little bit about gene regulation. So we know that genes are those sections of DNA that code for your proteins that give you your traits. So why do you have the hair color you have, the eye color you have, the blood type you have? Well, it's because what sequence of nucleotides you have in your DNA are dictating what protein your cells are making, and that is determining your phenotype. So for example, I have brown hair because inside my DNA is instructions to make the pigment for brown color inside of my hair follicles. That protein then gets made inside my hair follicles, and ta-da, you look at me and I have brown hair. What we find, though, is that the actual instruction piece of your DNA only accounts for about 1%. And now we kind of need to take time out right here to realize how wild that is. We think that the entire reason that DNA kind of is important is because it contains the instructions for making these proteins. But when the Human Genome Project looked at all of the A's, T's, G's, and C's inside of a human cell, they found that only 1% of those A's, T's, G's, and C's were really instructions, direct instructions for making a protein, which then begs the question, oh my goodness, what's the rest of it? Why is the rest of it there? And what we found, and this is still a huge field of biology right now if you're interested in this, is figuring out sort of what's going on there, is that the vast majority of those directions that are inside of our DNA are actually regulatory instructions. So what we find is that within the genes, there's actually different ways that those sections of A's, T's, G's, and C's can be cut or spliced together to make different variations of the same protein. And about 25% of our DNA is actually different splicing or regulatory instructions to put these different pieces together. Essentially, what we're going to look at right now is sort of how does that mechanism work. So we're looking at sort of how the gene is set up. So before you have a gene sequence inside your DNA, you're going to have what's known as a promoter region. It's also called a TATA box. That's because you guessed it, yeah, it has TA repeating in it. So thymine, adenine, so hence TATA box. And essentially what this is it's a little beacon. So you have a molecule called RNA polymerase, and you're like, that sounds a lot like DNA polymerase. And I would say, bingo, you got it on the nose. DNA polymerase does complementary pairing during replication, RNA polymerase is going to do complementary pairing during transcription. So it's going to pair A's with U's, it's going to pair T's with A's, G's with C's, and C's with G's. So that's what your RNA polymerase is going to do. But where RNA polymerase, how does it figure out where to start copying? And how does it know within all of those letters of the DNA what's something that needs to be copied? That's what the purpose of a promoter region is. It essentially acts as a little beacon for the RNA polymerase to know this is where you start copying. It's then going to copy, so this is your RNA polymerase, so it locks onto that TATA box, that promoter region, and then it literally just moves down the sequence of DNA, and it's going to do that pairing, A's with U's, it's going to do T's with A's, G's with C's, and it's going to create that molecular photocopy. You can see down here a nice kind of simple diagram of what that looks like. So we notice that our DNA splits again, RNA polymerase sort of moves down the strand, but instead of us making two complementary strands, so having that leading and lagging strand like we saw in replication, you notice that just one strand is going to act as the template on our double-stranded DNA, and that is the only side that's going to get converted into an mRNA strand. Now, your cells all have kind of the master copy for all of your genetic information in them. So kind of one of the things we know just from having lived life is that different cells in your body look different. So if you picture in your head right now a neuron, so a cell you might have in your brain, and if you picture a red blood cell, and if you picture a muscle cell, if you picture a skin cell, you're like, oh my goodness, these things look really, really different. And the reason that they can look so different is because although they have the same genes in them, so they technically have all the same instructions in them, they're not all using the same genes. Your body actually has the ability to silence certain genes, so essentially flip them off. Like, for example, you are not going to necessarily need to know how to make hair color protein inside of a white blood cell. Your white blood cells don't care what hair color you have, but do they have the genes for your hair color in them? Yeah, they do. They actually do. So what your cells have the ability to do is to silence promoter regions. And how they essentially do this is they stick a methyl group. And so a methyl group is just a simple kind of a CH3 molecule. So it's a really tiny one. But when that methyl group sits on that TATA box, it essentially makes it so it's invisible to RNA polymerase. It's like RNA polymerase can't see the gene that it's there, and it effectively silences it. Now it's still there. So if you're asking, hey, could we, if we were geneticists, could we look at people's white blood cells and could we tell what hair color they have? Could we tell what blood type they have? Could we tell all this genetic information? And the answer is yeah, you can, because you can still read the codes that are in there. It's just that the cell isn't paying attention to those genes because they have been silenced. They're not using them. So one of the things I was talking about earlier is about how your DNA is only 1% proteins and the other 25% is all kind of regulatory mechanisms, so different ways to splice those proteins. And this is essentially getting into that. So not all DNA is transcribed. Once mRNA is made, so your RNA polymerase is going to come in, it's going to attach to that promoter region, it's going to make a long strip of RNA. So it's going to go all the way to the terminator region, which is what tells RNA polymerase to stop. And you'll have this really long strip of mRNA. However, it's not done yet. What's then going to happen is what's known as RNA splicing, meaning this RNA strand is going to get chopped up into sections, and some of those sections are going to be called introns. Introns are sort of the non-used pieces. They're going to stay inside the nucleus. They're not going to be used to actually code for the protein, and it's only the exons, so the things that are going to exit the nucleus and head to the ribosome, that actually gets used. Now, that 25% of your DNA, what we're finding more and more, so this is much more like modern biology, like in the last 7-8 years, is that a huge portion of our DNA is actually varied instructions for how to cut mRNA. So how to make different versions of the same protein just by cutting different places and making varied introns and exons. So as we said, so here's kind of a visual of what I'm talking about. So here's your DNA. So we notice that inside the DNA itself, you have intron and exon regions. You get your initial transcript. So RNA polymerase comes down, it makes one long strip of mRNA, and then you would have your splicing. So this means that enzymes come in and they're going to chop out certain regions of mRNA and kind of garbage can them. So your introns are they're garbage canned. Nobody's going to look at those. They're just going to break back down and be reused as kind of like raw materials to maybe build another RNA strand. And then your exons are going to get sort of stitched back together to create your final mRNA sequence. And again, what I was saying here is that we're finding that a huge portion of our genome actually is coding for different ways to do this splicing, so to create different proteins even from the same initial transcript of mRNA, which is pretty cool. So we did this one. So our DNA polymerase is the enzyme responsible for assembling nucleotides and replication. What do we think assembles RNA nucleotides? Good. So hopefully you guys were able to get that this is going to be your RNA polymerase. So polymerase literally means enzyme that makes a polymer, makes a big molecule. If it's DNA polymerase, it's making a big DNA molecule. If it's RNA polymerase, making a big RNA molecule. So summary of transcription, kind of what we've just seen, is that you have your DNA molecule and it's converted into a single-stranded mRNA molecule. It then is sort of spliced together to create that final mRNA molecule, and now it's ready to leave the nucleus and go to the ribosome. So this is where we're actually going to do translation. We're going to take that coded message that we essentially photocopied in the first half, and we're going to turn it into a protein. Before we can do that, we need to make sure that we understand a little bit about protein structures themselves. Now, you guys have this, so you'll notice that yours are blank lines, so you just need to make sure that you're filling in the labels on here. Proteins themselves are actually kind of made up of two subunits that sort of sit together, almost like an open clamshell. Like if you can imagine in your head, kind of like a clamshell on the beach that's sort of half open. And they have a large subunit and a small subunit. And when these attach, you're going to end up with 3 points on it. So your large subunit is going to open up a little bit. And those are called the E site for exit, the P for the peptide sites. This is where you're going to make bonds. And then you have A. Oh no, my bond. So you guys are going to have to do this just by listening to me. So it's E, P, and just like the environmental organization, A. So this last one right here, I don't know why my writing isn't popping up. It's called the A site. That's the attachment site. And we're going to watch a video of this in a second, but right now we just want to know the anatomy or the structure of a ribosome. So large subunit, small subunit, and then 3 binding sites right there called the E, P, and A site. And this is kind of showing you a nice little preview of what's going to happen here. We're going to see that mRNA, so that single-stranded photocopy that we made is going to kind of move through those EPA sites. We're going to build our protein off of here. It's also at the ribosome that we find our second kind of appearance of RNA. So we've talked about RNA as being kind of this essential supporting character. You can't have our story of protein synthesis without it. So we still have one version of RNA acting as a messenger, so actually doing the copying of the gene, being spliced up to form a final sort of recipe for a protein, and then leaving the nucleus. And here we see our second sort of appearance of RNA. A different type of RNA called rRNA, called ribosomal RNA, actually makes up the ribosomes themselves. Ribosomes are actually assembled inside the nucleus. You have a special region inside the nucleus called the nucleolus. You may have seen this sometimes, like in a diagram of the nucleus, there'll be like an even darker region. An even darker region inside the nucleus, and that's your nucleolus. It's where your cell essentially is putting together these ribosomes, and it uses RNA in order to actually physically construct them along with other proteins. And again, totally essential because you have to have ribosomes in order to put proteins together. And we'll look at kind of, they physiologically are going to help you kind of put these molecules together. We need them. All right. So quick view of where we're at. So we saw DNA being converted into mRNA during transcription. So that worked by pairing A's with U's, T's with A's, and C's with G's. And then we spliced that RNA together and we got our final transcript. And now we are sort of talking about how at the ribosome we just discussed, we are going to convert that RNA into a protein sequence. So mRNA, kind of picking up where we left off, exits the nucleus and travels to the ribosome. It then connects to the A site or the attachment site on the ribosome. So you would essentially have your RNA moving in and attaching to that attachment site. So there's directionality when it comes to the movement. It's going to go A site first for attachment, then P site, then E site, which is your exit site. In terms of where this RNA is actually heading, well, it's going to depend a little bit on the type of cell. So we know that they're going to ribosomes, but if we kind of were to try and picture in our head, well, where are these ribosomes? It depends a little bit on the cell again. All cells are going to have ribosomes in their cytoplasm. So sometimes this RNA is just going to leave the nucleus or leave sort of the region with the DNA. And if we're talking about a prokaryotic cell, and it's just going to go into the cytoplasm attached to a ribosome. However, if we're talking specifically about eukaryotic cells, so cells like the ones that make you up, you also have a second place where ribosomes are attached, and this is the rough endoplasmic reticulum. The rough endoplasmic reticulum is a continuous sort of maze of membranes that are attached to the nucleus, and the rough endoplasmic reticulum has ribosomes embedded into these membranes. So sometimes what will happen in a eukaryotic cell is the RNA will leave the nucleus, and then as it's traveling down this endoplasmic reticulum, it'll just sort of attach to one of the ribosomes that's embedded right into the wall of the endoplasmic reticulum, and translation will occur there. But again, that's only eukaryotic cells. You are not going to find an endoplasmic reticulum, as that is kind of a membrane-bound organelle in a prokaryotic cell. The last one, or before we look at translation, is we need to kind of have a discussion of our last piece of RNA, and that is tRNA, or transfer RNA. Transfer RNA is essentially the rock star here when it comes to actual translation, so converting that RNA into a protein. And that is because if you look at the structure of a tRNA molecule, it's absolutely beautiful for being able to kind of do that job. What we find on one end of a tRNA molecule is we find a sequence of 3 nucleotides. And this is RNA, so that means it is going to be A's, U's, G's, and C's. But you're going to find on these RNA molecules that they have a unique combination of 3 proteins, 3 nucleotides on this end. And what we find on the other end is that they have a corresponding amino acid that they carry with them. So this is the really, really cool thing about tRNA. Depending on what sequence of 3 nucleotides it has here, that's going to dictate what amino acid it carries on the other end. And what you're going to see very shortly when we actually look at translation as a process is that it is this structure of tRNA that actually allows the cell to convert the message in mRNA into a protein. So what happens? So the green structure right here is your ribosome. This bottom sequence right here is your mRNA. So this is your sequence that would have just come from the nucleus. And these blue structures up here with 3 letters on the one side and a box with kind of a short abbreviation for an amino acid on the other side, this right here, that whole thing, is a tRNA molecule. So what happens is the mRNA sequence is going to attach to the A site, so the attachment site of a ribosome. And the first 3 letters are going to get read. So in this case, it would be AUG. And these first 3 letters are called a codon. So it is literally a coding region of mRNA. Now, in order to figure out what amino acid, so what part of a protein these 3 letters are asking for, the cell is then going to use complementary pairing between mRNA to determine what amino acid needs to come over. So AUG is going to be complementary to UAC. So we're going to notice this is really similar to what we did with DNA replication, but hey, here now we're doing it with complementary mRNA sequences. So A's with U's, G's with C's. Now, if UAC is on one end of a tRNA molecule, then methionine, or METH, is the amino acid that would be attached on the other end. So that means, kind of imagine this, you would have this tRNA molecule with UAC on one end and a methionine on the other end just floating around inside the cytoplasm. As soon as this AUG sequence pulls up to this ribosome, the cell is essentially going to kind of take this out of the cytoplasm, realize that this UAC kind of pairs up perfectly right here through bonding. They're going to know that methionine is the first kind of amino acid that needs to get built. And then the mRNA is just going to slide. So remember that this would happen kind of at your A site, and then it's going to move through to the P site, and the next 3 letters in your mRNA, or your next codon, is going to get paired with your next 3 letters of your tRNA molecule, and it's going to bring over a new amino acid. So if my next sequence was CAG, complementary pairing on the tRNA would be GUC, and it would bring over a glycine, or GLY. And this process is literally just going to keep happening. So mRNA keeps sliding through. The next 3 letters, or a codon, is paired with 3 letters on a tRNA molecule, or an anticodon, and a corresponding amino acid is brought over. And literally what's just going to happen off the top of our ribosome is you're just going to get a chain of these amino acids that are going to start built off the top here. And the mRNA will just keep shuffling through until it gets to the end. And then the ribosome will realize that that is the end of the kind of reading sequence for building the protein, and it will release sort of this sequence of amino acids that it's assembled on the top right here.